Okay, good. Thank you very much. A number of affiliates coming. Yeah, I'm glad to see everyone in person. So welcome to single pluripotent stem cell handling and cloning workshop. This one is a very unique workshop. So therefore it's very special. And also part of this one is stage one stem cell education program supported by some grant we recently received. And also, just to introduce myself, I'm Gitai Kim, the director of the UCLA Human Stem Cell and the Genome Engineering Center. And also thanks for your participation. Some of you already joined the basic stem cell training section. We handed about 150 registered. The training was really joined, and then we had a wonderful time. And then we just continue to this success through training. Okay, and let me briefly introduce what is the UCLA Human Stem Cell and the Genome Engineering Center. And usually we call UCLA HSCGEC and the GEC is GEC. And we support research in iPSC reprogramming and the genome engineering and also quality control using the unique part is robotic automation. And our center is led by Dr. Cho Kyung-cha over there. And she is leading the stem cell team. And also Dr. Deniz Ata is here. Could you see? Yeah. And he is leading the genome engineering team. So recently our center awarded about $4 million from the some infrastructure grant and to expand robotic automation. So if I already gave the warning to Ryan because my laboratory area is really messy because a number of equipment is installing. And hopefully we relocate the bigger space to accommodate all of the client need. And this equipment can generate not just 1 digit or 10 digit number, 100 digit number, something 100, 200 iPSC generation, or 100, 200 gene of the gene editing by the robotic arm. So this is the really great opportunity. Just to think about what if I can generate 100 gene editing and 100 iPSC isolation. Just to think about it. Bring up the idea, suggest your lab head, and then we can collaborate. And also we can submit the grant together. We are here to support your research activity. And today's workshop is organized in partnership with Biotechnic, whose the single cell sorter is also available in our center, and you can access this equipment through us. And then also Biotechnic is really leaders over this field, and then they are fully support your research. So therefore, do not hesitate. All of the staff member, Kati here, Ryan here, and then get the best support you can have. And now it is my pleasure to introduce Dr. Ryan McCobbs and from Biotechnic, who will lead today's program with a multiple presenter. When he come here, just with a big crowds and then shouting and then just welcome them.
So thank you all for coming. My name's Ryan McComb. I'm the product manager for this instrument here called Apala. I've spent the last 2 years of my life kind of really focused on this, and we're really focusing on the stem cell and cell line engineering market for this instrument, and you'll hear about it. But I just wanted to first start off with just some logistics about how today will run. So we do have some people on Zoom that are joining virtually, but we're going to have 4 presentations. The first is going to be Dr. Deniz Ata. The second will be from Kadi Tormanen, who's our field application scientist. And then the third will be virtual for Dr. Evi Hegbish, who focuses on organoid and also differentiation products at Bio-Techne. And then the final presentation will be myself. I'll talk a little bit about iPSC stem cell work that we've done at Bio-Techne. And then we'll have lunch delivered here. And there's going to be plenty of food. So don't, we're at max capacity, but we have enough lunches for everyone. And at lunchtime, we were going to take people into the lab to do some basic, walk through the instrumentation. Dennis will be giving you a tour of the lab and the workflow and the instrumentation they use. And Kadi will be doing kind of a live demonstration with iPS cells on the Pala. And then we're going to have to kind of break it into groups. So, and one other kind of wrinkle is we actually have to be out of this room at 1 o'clock because there's a group coming in after us. But what we can do is we can have people going through the lab and maybe if you want to stay late after 1 or to 2 or later to kind of go through that, we can accommodate that as well. Bathrooms, you pretty much just walk in either direction, right? And you'll run into a bathroom and eventually. Please ask questions through the presentation. We're gonna have this as kind of an interactive session. We're just grateful you're here and please, let us know if there's anything you need. And we think everything will go smoothly. We've got everything set up pretty well. And we're first going to hear from Dennis. So remind me how to pronounce the university you did your PhD at. Koç University in Turkey, studying neurodegenerative diseases. And he came to UCLA to set up the lab here with Katai Kim. And is the senior lead research associate. And so without further ado, we'll turn the time over to Dennis. So thanks.
Is it ready if I sit? I don't know. What? Should be good. You're good. You're good. Okay. So hello everyone. Welcome today to our workshop. Today we will explore how semi-automation can transform workflows. We will first look at what are the challenges of manual work and manual gene editing workflows. And in this session, there will be a short walkthrough of gene editing workflow. At noon, you will see a portion of that workflow in person. And we will also go through why we automate, how we automate, how we do the design and Cas9 knockout gene editing workflow in our labs. So first, so why we automate? Because first, it's time consuming. A lot of tasks simply can take too long. Manual workflow eats up a whole day. And it takes even more time if you're working in GMP environment. And automation helps you cut down the precious time you have. So this way you can focus on your work other than just doing repeat tasks like plate coating, medium change, and passaging. And second, it's error prone. Repetition is where the mistakes are creeping in. You'll be pipetting the same volume hundreds of times. We will be transferring lots of cells from one well to another. And it's really easy to mess up as you get tired because you can start to lose your focus.
And tips from multi-channel trends sometimes just like don't aspirate well, sometimes fields fall over, and those can also mess up an experiment really a lot. Automation doesn't eliminate errors entirely, but it reduces the human slip-ups that naturally come with repetitive work. So also manual work is highly variable. Even when nobody makes a mistake, manual work is rarely identical from one person to another. Even a person, even one person in the morning versus the same person in the evening can do slightly different variations in hand motion, which could lead to variability. And of course, like between interperson, effect is even more higher because one technician might pipette faster, one technician might be holding the pipette a little bit slanted. So those small differences can grow into big differences in the data. And automation gives you consistency. Like you will always get the same action, same way every time, which makes results more comparable and reliable. And of course, at higher scale, it's harder to do the workflows. What might be manageable for a couple of samples or a single plate becomes quickly tedious work, overwhelming work as you try to scale it up. More plates mean more time. It opens more room for errors and more variability. In general, in our lab or any other trained person can handle 5 stem cell lines simultaneously without having problems with synchronization, because when you have 5 different stem cell lines, it's hard to synchronize them. Some of them will be confident at Friday, some of them will get confident on Sunday. You will have to come work over the weekends all day. So automation, like with automation, once the workflow is set up, scaling becomes much easier because there are no errors, no mistakes, and you can process more samples without multiplying the problems. Like you can increase cell line per person from 5 to 50 with ease because the machine will be doing the tedious job for you. So how we automate? We automate through practical integration. There are 4 rules we follow to make integration work in practice. The first one is know what you do. Because before you try to automate something, you really need to know the process yourself. Because if the manual workflow you're already doing is messy and inconsistent, machine will repeat the same mistakes or follow the same pattern. And if you don't fully understand what you're doing or don't familiarize yourself with the protocol you're doing, it will be very hard to fix those mistakes. So before trying to automate something, spend time to make sure the manual version is clear, reliable, and you're familiar with it. And second is start simple. Just like the long-term vision is a fully automated system with adaptive protocols powered by AI, but you don't get there in like just one jump. You should start with repetitive, tedious work, error-prone steps that take time, like coating 96 file plates, changing medium of 96 file plates, stuff like, or single cell sorting your samples. Even automating these steps will save you a lot of time, and it is going to show immediate value. And validate incrementally. So don't try to flip the whole workflow overnight. Add an automation step, do it step by step, and check each step you automated versus the manual workflow. Run side-by-side comparisons, catch issues early, and it's going to be at first, it's going to feel more tedious than doing the actual manual work itself, but once you do it, you will feel very good about it, and it's going to save you a lot of time and money. And design smart, but also be realistic. One of the few things you actually have over full control is the design you do, because after you start your experiment, you will have less and less control with every step. So it's important to design before you start your automation process, because also designing smart doesn't mean you should chase extremities. Like don't try to do like most flashy newest approach. It's about like choosing simple formulas that can that can work and just build slowly on it. Like find something that works, replicate a working formula and build on it. It can be like picking a commonly used validated plasmid backbone or sticking with a standard promoter. Just adapt the working formula to your workflow. So again, we have again, the part we actually have the most control is design. I want you to show you a few design tricks that can help automate your workflows. First one is to simplify your design. For example, like in reinventing the whole plasmids every time for each project, you can build a standardized backbone which you can adapt to all your gene editing projects. For example, you can generate the standard selection cassette, and you can have a standardized backbone. So whenever you want to report to cell line generation, maybe you will receive a project for like 20 cell line generation. With this design, you can simply just order these homology arms from your favorite oligo provider, and you can just anneal them quickly. This way, you just save time from like design and procurement. With this kind of approach, we were able to cut down our repair template generation from like 1 month to 10 days. Because now we just simply order these arms, whatever the project is, and we just anneal them all together. So it takes roughly around 10 days. Another example of simplification can be seen in this example. Normally, conventional reporter, this is for our reporter. This is one of our reporter templates. Conventional reporters usually have like a P2A here, followed by a fluorescent reporter and followed by poly-A sequence. We basically, and those usually have like a big chunk of selection cassette just after the actual fluorescence protein. In this design, we took the selection cassette and put it just before the whole thing, because this way, this EF1 alpha promoter can also activate this fluorescent gene. So this way, we save we save from the size, and by switching from P2A to iris, we standardize the design. So this way, we don't have to check for like if it's in frame or something. We don't have to manually modify it, because this way it will be standardized. Another design changes we did it here is we removed poly-A sequence, because even if it's low, even if there's a low chance, there's still a chance of random integration. So by removing poly-A, we destabilize this construct. So it will increase your chances of selecting an actual insert, because this design will be using the endogenous poly-A sequence. So if it randomly integrates somewhere out of a gene, it's not going to be giving a signal or being resistant. Also, if the cargo size is still an issue, you can just simply remove the promycin here and like make it even smaller, because usually when you try to do an insertion, efficiency lowers significantly if you try to insert something like 4,000 base pairs. With this design, we were able to minimize it down to about like 2,800 base pairs. Also, thanks to our single cell sorter, we don't need to do an antibiotic selection. We can just transfect the cells and sort the cells for the green signal, because it will be active because of the EF1 promoter. Then we can remove this cassette. Then we can sort the cells. Like first, we can sort for the positive selection, doing RFP positive sorting. Then once we remove this cassette, we can do RFP negative selection this time. And when you eventually differentiate your cells into your desired cell line, they will be red again. In this case, this approach is what we call on-off on reporter cell line system. So this way, without the antibiotic selection cassette, you can minimize your cargo load and you can achieve success. So today we will have chance to conduct single cell sorting in our in-person training, but I want to quickly go through the parts that you're not going to see today. And this is basically I will basically go through our. Maybe I should say. We will go through our knockout workflow. We basically start with designing 3 guide RNAs. we design 3 guide RNAs that are spanning roughly 150 to 200 nucleotide region. We usually prefer a region just after ATG or exons with major isoforms of interest. Things to consider here are there might be different isoforms you might want to knock out, sorry. So you need to study for that before you start. Again, it's part of the designing. And you should pick guide RNAs with high efficiency, like high efficiency estimates and low off target score. And you should also make consider if this gene you're trying to target is crucial for stem cells, or if they are if you're working with cancer cells, knocking them out very early can lead to cancer cells acquiring tolerance to that knockout, so you may not see your phenotype and it will affect your results. So you should all consider these kind of variables. So the first step of our experimental step is electroporation. Our success criteria here is reaching 70% transfection efficiency, but since we don't have any color reporters here, we always want to control electroporation. In this case, like an RFP, just a simple RFP plasmid electroporation. The control group tells us whether electroporation worked and whether the cells can tolerate electroporation process itself, because sometimes it depends from cell line to cell line. They don't like it, so we have to do some minor adjustments so the cells can tolerate it. And the second part, which we are going to see today, partly, is expansion and IC analysis. I will go through in details of IC analysis. It's called it's inference of CRISPR edits. I will briefly mention. I will mention in detail in the next slide. So after electroporation, we move the cells into 12-well plate because this is to minimize the time needed to reach confluency. Because this is important because we don't want wild type to take over the plate, because there might be a selective pressure because of our gene edit. So this setup also gives us flexibility. We can sort one well, we can freeze the other and isolate DNA from the other well to do IC analysis. Yes. We deliver Cas9 as an RMP. So we deliver Cas9 protein coupled with 3 guide RNA. We first mix them in a Pandorf tube. We wait around 10 minutes for them to form complex. Then we add that to our electroporation mixer.
How do you measure all of their work if those are, oops, sorry. And so what's the efficiency?
Efficiency change, it differs from experiment to experiment. We make sure we do at least 3 replicates for each experiment. We check electroporation efficiency by having a control group, like RFP control group. So this way we can assess what's the efficiency rate. If it's less than 70%, we just don't continue with that batch and move on with the other 2. If it's 70% or higher than 70%, we just move on to the other step. So again, after moving to 12-well plate, we freeze some of the cells because it's important to freeze them as early as possible, because again, we don't want wild type to overtake the wells. And we isolate DNA to do IC analysis, which is also another success criteria for us, because we will be able to see what's the indel score, what's the gene editing efficiency. For that stage, we want to have 70 to 80% indel efficiency, because we want our life to be easier after that point. Because if it's too low, like 30%, it becomes like a game of chance, because you can sort 100 cells and maybe you're not going to see an edited cell after the reads you do. And following the population ICE, we sort the cells using our Pala sorter. Then we grow them. Then we grow the cells and we do IC analysis again doing on the single cell clones. So we do IC analysis, like I said, it's called inference of CRISPR edits. We basically PCR amplify our gene of interest, and we compare the relative contribution of each sequence read of mixed population versus wild type. So I want to show you one bad example and one good example of reads. So in this example, it's indel percentage is about, doesn't work. Indel percentage is about 35%, which means 65% of the reads are wild type. So this is not good for us, so we don't move on with this one, because wild type read is too high. Probably most of the cells you clone will be wild type. And on the second example, indel ratio is 100%. This is usually what we achieve in general. And also the top read is deletion of 137 base pairs, a nucleotide deletion. And it's also causes a frame shift. So we usually consider this kind of knockouts because we also want, yes, we want to delete a big chunk, but we also want to cause a frame shift just to make sure everything is smooth. Because again, this is also like part of the designing section, because after that, we don't know if they're like unknown isoforms or another ATG will form. So we just cause a frame shift. And then we grow our single cells and we do another round of IC analysis. As you can see, this time it's 100% reads. Again, but only reads are coming from 137 base pair deletion. So then we do our basic pluripotency staining. We do semi-analysis for chromosomal abnormalities and stability, and we wrap up the project. So thank you for listening. I hope I was able to go through it without any problems. We will see the cell sorting stage. This stage, you will be seeing from 12 valve to 96 valve plate stage in person. But if you have any questions about the other stages, you can come find me. You can always reach me out from through my email, which is very super easy. It's data@mednet. So it's easy to remember. Thank you all for listening. See you in our afternoon session.
Better we'll switch up here.
We have some time.